Hey everyone, my name is Xander, and this is my local expert project on Cryo-Em. So Cryo-Em is cryogenic electron microscopy, and it's a way to study structural information of tissues, bacteria, viruses, proteins. We're probably all familiar with normal microscopes. Those use the visible spectrum of light, so that's anywhere from 400 to 700 nanometers. And that's fine if you want to view things larger than 400 to 700 nanometers. Human hair, for example, you can see that under a microscope. You can get an aqueous solution and see a larger bacterial organism. But what if you want to see a virus at 100 nanometers or a protein at 10 nanometers? You're going to need something smaller to get the resolution that you want. And that's where electron microscopy comes in. So with electron microscopy, you have electrons that are accelerated down a vacuum. You can see that here at different voltage ranges between like 80 and 300 kilovolts. And the scattered electrons, so it passes through your specimen, and then the scattered electrons are refocused on a lens, and that's what you end up seeing. Now, when they're accelerated at 300 kilovolts, they have a wavelength of 0.02 angstroms, which is 0.002 nanometers. 1 angstrom is 0.1 nanometers. So it's great for seeing things that are super, super small. It gives you the resolution that you want. The problem with normal electron microscopy is that electron radiation damages organic tissues. It breaks down the chemical bonds, it destroys your DNA. So that's where Cryo-Em comes into play. Cryo-Em is using liquid nitrogen or liquid helium, and it's freezing your samples, as you can see here, in a glass-like structure. And doing this actually reduces the radiation damage about sixfold. So you're able to view things that you otherwise wouldn't because your electrons are just degrading your sample. One application of cryo-EM is cryo-electron tomography, and that's what you can see here. So your sample, as the electron beam is passing through your sample is, and you can see it here too actually, is changed to be at various tilts. And this lets you take images from different perspectives of, whatever you're trying to look at, a protein, for example. And then you can use software to create a composite picture, taking all these different tilt pictures into account to get you, a full picture of what's going on. Now, in 2013, this method was called blobology because, as you can see, you don't really get an idea of what's going on here. It's all mashed together, and you can't really get anything useful from this. But the resolution at present is actually atomic resolution, so you can see the actual atoms and the alpha strands of the protein, and I'll show some of that later. But it's come a long way. So these are two figures that I found. On the left here you have bacteria, and this is actually showing the interior of a bacterial cell. So A here is just what a bacterial cell would look like, and then B is your 3D surface rendering after taking all those pictures together and creating something in software. And then C, this is showing higher magnification. These dots here are actually ribosomes. And then D is showing chemoreceptor arrays, and that's what they were studying in this article. How bacteria use these chemoreceptor arrays to move through their environment based on concentration gradients of molecules, and then they could use these cryo-EM pictures again at various tilts and what have you to piece together this picture to understand how these arrays actually how they actually look. One other thing here, this is actually the nucleoid. It's where the genetic material is stored in this bacteria. And then on the right, we have Mayaro virus. One thing I want to point out was this link, actually. It's a pretty cool link. It goes over Mayaro virus. It's a dengue-like virus, and it talks about the class of virus, is it an emerging threat, along with the different variations of virus in its family and the symptoms that they produce and the life cycle and the incubation time. It's not a super dense article, so it was pretty easy to read, but I thought it was pretty interesting. I'll I, I'll probably put this in the comments, actually. I don't know how else people would get to it. But yeah, check that out if you're interested. So the figures, though, what they were doing was they used Cryo-Em to actually image this Mayaro virus. And C and D, these are just cross sections, but A and B are the actual different images. So A is basically a heat map. It's a resolution map of the angstroms that they actually got while they were taking images. And so the red is 5.6 angstroms. It's a little, less, it's lower resolution, but 5.6 is still pretty good. And then your blue here is 3.7, so you have pockets of super high resolution. The global resolution was actually 4.4 for this entire virus. And that's really good because when you go over to B, you're able to see protein structures, how everything actually comes together, how it fits. And this whole thing is showing that the spike protein of this Mayaro virus is a trimer of heterodimers, and it lets you get that information of how the spike is created. We talked about glycosylation in class, so it has post-translational modifications on it as well. So, but it's just good to understand the structure of your virus. Okay, so module tie-in in class we've talked about Western blots and immunofluorescence and ELISAs, and all those are good for what they do. So Western blots and ELISAs, you're kind of asking the question, is my protein there or not? With ELISAs, you're using conjugated antibodies to get a signal with your substrate, so maybe some horseradish peroxidase, and then you're changing a color, you're answering, yes or no. Western blots, you're getting molecular weight information as well. Is my protein the right weight? Is it degraded? That whole thing. So do I have my protein? And then immunofluorescence is telling you some localization information. So you have your actin staining here in red, you have your DAPI staining of the nucleus here in blue, and then maybe your green here is your protein of interest. So it's giving you some information about where this protein resides in the cell. Cryo-Em kind of fills in the picture here because you'll ask yourself, okay, do I have my protein? Yes or no? Where is it? Okay, I can see where it is, but then how does it actually function? And that's what Cryo-Em is asking. So this was a study that was in the early days of the pandemic, looking at SARS-CoV-2, COVID-19, and it was looking at the COVID-19 receptor binding domain and ACE2 interaction. So this is the receptor binding domain on COVID-19, and then this is your ACE2 protein. And it had a total resolution of 3.5 angstroms, so 0.35 nanometers of resolution. So they were really able to understand what the interactions were between the virus and the proteins in our cells. On the top here, you could see ACE2, along with its protein complex. ACE2 sometimes forms in a complex with this beta AT1 transmembrane protein. And then you see it from the side here, but the bottom is the really cool part, and it's showing the actual alpha helices and beta barrels and beta strands and their interactions, their polar interactions and nonpolar interactions between themselves and the COVID-19 receptor binding domain. So this is one of the main alpha strands, actually, alpha helices responsible for allowing COVID-19 to bind. It's this A1 helix, and you can see there's a number of polar interactions here. And then they were also able to look at hydrogen bonds and van der Waals interactions using this method. So it gives you the resolution required to see things on this level. What are the actual interactions that allow COVID-19 to attach to ACE2? And the whole takeaway from this information is trying to design therapeutics, antibodies that can suppress viral infection. Maybe it's messing with this interaction here or preventing this interaction. So controls, One of the problems, one of the limitations, I should say, with Cryo-Em is that your sample has to be super pure. If I go back here for a second, this I said is a glass-like substance, and it's important that it's glass-like. If you have imperfections here and ice crystals, that's really going to mess with the imaging software, because you're trying to take these composite images, and then now if you have, ice shards in there and you can't see, it's going to really screw that up. So it's super important that your sample is pure, and that's why you're doing a lot of purification controls and then a negative stain microscopy. And I actually said that here. So yeah, products can interfere with computational analysis. That's the whole thing that you want to avoid. So one thing you'll do is an SDS page gel. You're looking for right molecular weight, you're looking for degradation, do I have the right protein? Is it not degraded? Okay, good. And then maybe you'd move on to some size exclusion and affinity chromatography. So you're going to run these a few times because, again, you want everything to be super pure. Maybe you're using a buffer exchange at the end, some dialysis membranes, and you're really trying to just isolate what you're interested in. And then once you think you have a pure sample, what you can do is negative stain microscopy, and that's here at the bottom. And people refer to this as the gatekeeper of cryo-EM. It's cheaper, it's the equipment is more common. It lets you see about 20 angstroms at best, so you're not getting the high definition resolution that you would be getting otherwise, but it's a lot cheaper, and it's a quick way to just check that your sample's good before cryo-EM. So you're going to use different substances here, uranyl acetate or formate, and then some others as well as staining agents. And those staining agents, what they're going to do is they're going to paint your background here black, keeping your biological sample white. And it's kind of like black paint against light. So black paint's going to absorb all your light. It's not going to reflect. Your sample, on the other hand, is going to reflect the electrons. So you can see any biological samples if there's any impurities, whereas your background is going to be all blacked out. And then if that passes, then you can move on to cryo-EM. Okay, so cryo-EM versus X-ray crystallography. X-ray crystallography historically has been the gold standard. You're able to see atomic resolution with X-ray crystallography. The main advantage, though, of cryo-EM is that crystallization is not required, and that's the biggest pain point for X-ray crystallography. Protein crystallization can take months, years. Some proteins just never crystallize, whereas cryo-EM, you could take images and then you have a week of data collection and then a nice computer to spit out your composite image. When crystallized, the other thing is you can only view proteins from certain conformations. So actually, I should back up. In order to crystallize, you need to form arrays of repeating structures. That's what a crystal is. And some proteins are super large. They have polar interactions, nonpolar interactions. They have acidic sections, basic sections. All of those resist crystallization, so it's hard to get them to crystallize in the first place. And then if you can get them to crystallize, you're only seeing that in one conformation because there's only one way that you can arrange that protein that allows it to crystallize. So that means you're only seeing one image. Cryo-EM is nice because you're freezing your image. You're freezing your whole substance, and then you can take your image at different tilts. So you can freeze it at one point in time where maybe the protein has one conformation, or another point in time where maybe it has another conformation. And it gives you more flexibility to see how the protein actually acts in nature. Um, oh, and then another advantage, it's not specific to cryo-EM because X-ray crystallography also has atomic resolution, but I mentioned earlier how cryo-EM used to be called blobology, and it's because you just couldn't see anything. So now, you're combining this atomic resolution with the fact that you don't have to crystallize your proteins, and you can view it from any conformation with tomography, and you can just freeze it and take images at different tilts. So this really lends itself well to being able to study proteins in their natural environment instead of forcing them into some sort of conformation that may not be natural or may not just happen at all. This is an image from one study where they actually achieved a 1.2 angstrom resolution, and I think that's the record now for Cryo-Em. You could actually see the individual atoms here that they came up with. So that's pretty exciting. And then limitations, as for a lot of things, cost is a big one. So this machine costs about $8 million US to buy, and then it's about 350,000 US to run yearly. Not to mention that it's highly technical, so you can't just have anybody, you can't just roll out of bed and operate the machine. You need to be trained. This is a pretty technical operating position, so you really have to know what you're doing. You need to have a lot of expertise, and the machines themselves are super expensive. And then the other limitation is it's just computationally demanding. So a full data set for a 3D structure like this can take anywhere between 4 and 32 terabytes of data. And then, not to mention the heavy duty computers that you're going to need to run them. So a lot of people probably going to use NVIDIA graphics cards, so you're going to have to max out your computers here. 256 gigabytes of RAM, NVIDIA 4090s. And a 4090 is just the top of the, I guess I would call it, residential market for NVIDIA, like what you can buy just as a consumer. But then they have commercial grade GPUs that are even more money. And then you have to think about how much power those computers are drawing, how much heat it's generating, and that's all just to create a nice composite picture. So the machines themselves, the Cryo-Em machines, are expensive, and then the computers that can actually create the images are also really expensive. And all that's something you have to think about. And then here are my references. So I hope you enjoyed, and thanks so much for watching.